Spring Harbor Asia. My name is Maoyan Chi. I am the director of Cold Spring Harbor Asia running the program here. Okay. So before I give the podium to Len, I would like to take this opportunity to welcome everybody, okay, and to thank you for your presence, and then give you a very brief introduction about Cold Spring Harbor Asia, the project, no more than 10 slides. Okay. So I think most of the audience knows Cold Spring Harbor Laboratory on the North Shore of Long Island. Okay. The laboratory has been a legend for life science, for biology. Okay. So because of time limit, I won't go through these details of those highlights of the laboratory. Okay. The point I want to make is that it's a legend, the laboratory, and we want to transplant this model, this legend, into this part of the world, in the Asia Pacific. Okay, and it is home for scientists. Okay, the particular culture Cold Spring Harbor Laboratory represents, we think it's a very highly valuable to the scientists in Asia Pacific. Particularly, it's a very relaxed. Okay, it's a it's a flat structure. Okay, you can challenge the authority, and you can share all the ideas bouncing with each other in the bar. Okay, or on the lawn, in the auditorium. Okay, so that's kind of exact thing we really want to replicate here in this very place. So 4 years ago, Cold Spring Harbor Laboratory, we started to thought about, to actually execute the plan to transplant this project to Suzhou. Okay, just give you a very brief, some idea about Suzhou. The town is about 1 hour from Shanghai by car. Okay, and we are actually to the west of, or to the east of the older town, which has a history of 2,500 years. Okay. And the Suzhou actually is famous for lots of its classical Chinese gardens. There are dozens of Chinese gardens located dotted in the older town. Okay. And we are in the new district of this city. Okay. I believe that we'll have a chance, or more than two, to visit the older town. Okay. And also, Suzhou actually is a home for classical Chinese scholars for the past 25 or 2 millennia. Okay. So it's a very interesting town, place to be. Okay. So this tells you, how this place was built. Okay. 2008, it was look like this, and by the late of 2008, the design was finished basically. Okay. Was approved, and the construction started a few months later in 2009 beginning, and it's looked like this in the mid of 2009, and we started the meeting program in April 2010, 2 years ago. So in fact, this very meeting marks the first of the 30 year. Okay, this is the very first meeting of this 2012. And we went through lots of different versions of the design. Okay, so here gives you a few sense of how things were laid out. And this was the note from Liz Watson, Jim Watson's wife, when the project was basically officially launched. Okay, so she wrote this note in a restaurant manual, in the back of a restaurant menu in Shanghai. Okay, so everybody was very excited. So I think it's interesting to just put here. So then, what we are doing here actually is to provide a very comprehensive and systematic scientific conference program in the Asia Pacific. Okay, so we are the third year of this program right now. And first year we had 11 meetings, and the last year we had 13. And this year, I'm going to have 18, and this is the very first of 2012. Another thing that we want everybody to have fun, okay, to really enjoy science and to enjoy science in a different magnitude, okay. Things can be very intensive, I know, during meetings from 9 to 9 in the evening, okay. But we also want to entertain everybody, okay. So also want to have science in a very stylish fashion. Finally, I would like to end my introduction by a quotation from Sidney Brenner. Here is the highlight, okay. Science actually is a social activity of the highest order, okay. So I want everybody to mingle together. I know this is Zebrafish is a very closed community, and you know what I mean, all right. And another thing, the last not the least, is today is our grand opening of the bar, okay, which is just outside of this auditorium on that corner. You can just folks can go there, okay. And another good news is that you can drink as much as you can for 50 yuan, which is $8, okay, for the 4 nights or 3 nights left, okay. Okay, thank you, everybody. And I'll give podium to Len. Thank you.
All right, well, that is good news about the bar, I have to say. I've had in the other Cold Spring Harbor, David Lane and I were talking, we had numerous times in the bar together, not only discussing in a friendly way, but also really having some issues out there too. So it should be very fun. So welcome to the meeting, Fishing for Answers, Zebrafish Models of Human Development and Disease. I should say this is actually the 5th time we've had this conference. We also call it Zebrafish Disease Models 5, and it's quite amazing. And pretty much every meeting has been roughly around the same 200 people. So it's been wonderful, and I hope that we continue this tradition forever. So thanks for coming all the way to China. It's great to be in China. We're very excited about it. Not only to just hear wonderful science and sit in a room, but I think there's a lot of new colleagues here, and we'd like everybody to interact. So we have certainly half of the audience is from China, and so we don't want to just interact with each other the way we usually do, but we want to interact with everybody. So I want to make sure that please introduce yourselves, come up to all the speakers, and make sure that we join as new friends. So with that, I'm very happy that everybody's here, and we're going to start the meeting. So we're going to go on to Disease Models 1, and our coordinators are Feng Liu and Liz Patton.
Okay. Actually, it's my great honor to be a co-chair together with Liz Patton, my old friend in Oxford a few years ago. So we start from the first speaker. Actually, it's Phil Ingham, who's from Singapore. He's going to talk about the sonic hedgehog.
Well, thank you very much, Feng, and thank you to all the organizers. For inviting me to speak at this meeting. I must say that when I received the invitation, I was delighted because I'd heard so much about Cold Spring Harbor, Asia, and I was delighted to have the opportunity to come here. It was a bit of a shock to then find I was giving a keynote lecture. So I've had to come up with something. So I thought I wanted to do something keynote lecture-ish, and it seemed appropriate for me to talk about Hedgehog since I've worked on Hedgehog for most of my career. And this seems to be a particularly propitious time to review what we know about Hedgehog. And obviously, I'm going to focus on our work on Hedgehog in fish. So I'm going to go straight in at the beginning, which was just to remind you of the fact that it's almost 20 years since Sonic Hedgehog was identified. And contrary to popular belief, Sonic Hedgehog was actually first identified in Zebrafish. Unfortunately, we didn't have the foresight to call it Sonic Hedgehog in the title, because I thought it was a silly name which would get forgotten very quickly. In a collaboration with Andy McMahon and Cliff Tabin, we set out to identify homologs of the Drosophila hedgehog gene, and we cloned these 3 different genes, these 3 orthologs of Drosophila hedgehog in the zebrafish, and this is the first one that we analyzed by in situ hybridization. And then we did an experiment which I don't think had been done before, actually, in the zebrafish, although, of course, it had been used very extensively in Xenopus, which was to ask what happens if you overexpress this gene. Of course, at that time, there were no mutants. Well, actually, there were 2 mutants. There was no tail and there was Cyclops, and that was it. So and we didn't have any way of making mutants, and we didn't really have a good way of knocking down gene expression either. So the only way we could manipulate gene expression was by overexpressing the gene. And so we took this approach of injecting messenger RNA, and that resulted in the upregulation of another marker, the only other gene that we were using in the lab at the moment, called axial, which we found was overexpressed. It was normally restricted to the ventral brain and the ventral spinal cord, and it became ectopically expressed. And so on that basis, we then had this idea that sonic was important in patterning the neural tube, and the data supporting that was also matched with data which came from Andy McMahon's lab, where they did the same sort of experiments in the mouse. And on the basis of those experiments, we actually filed a patent, which was not something I would normally have done. And in fact, my boss at the time, Paul Nurse, was at the ICRF, was very much against patenting scientific discoveries. But I think it's quite interesting because what happened was, as a result of this patent, this company was set up called Ontogeny Incorporated, which then went on to merge with 2 other companies in the Cambridge area to form a company called Curis. Okay, so the same year in 1993, so we hadn't just happened on hedgehog by chance, we'd actually been interested in hedgehog for a long time and studying it in Drosophila. In 1993, I think there were only 8 papers on hedgehog that had been published then. And we published this, which was the first hedgehog, the first form of the hedgehog signal transduction pathway, actually. And we came up with 3 variations of it. And the key thing here was this idea, which we'd published a couple of years earlier, was that the transmembrane protein encoded by the patch gene acted as the receptor for hedgehog. And this was this curious idea that hedgehog actually activated the pathway by inhibiting the activity of its receptor, which I have to say was met with a lot of skepticism at the time. Anyway, to cut to 3 years later, very excitingly, then 2 groups independently and simultaneously discovered that patched is a very important tumor suppressor gene, and that it in fact is responsible for the majority of these tumors called basal cell carcinomas, which are one of the most frequent types of tumor in Caucasians. And over the succeeding years, it's become clear that hedgehog signaling, or the aberrant hedgehog signaling, is implicated in multiple different cancers, some of which are shown here. This figure is taken from a review from 5 years ago by Lee Rubin and Fred de Sauvage. So this link between hedgehog signaling and cancer, which came via this discovery that patch is a tumor suppressor gene, then really initiated a search for chemical modifiers of the hedgehog signaling pathway, with a view to discovering molecules which could inhibit the pathway and then therefore could be used as leads for drugs which could be used to treat these tumors. And excitingly, just a couple of years ago now, Fred de Sauvage and his colleagues, in a collaboration between the pharmaceutical company Genentech and the small biotech company, which I mentioned a few minutes ago, Curis, developed this particular hedgehog inhibitor, which they, in phase 1 clinical trials, was found to have remarkable effects in causing regression of metastatic basal cell carcinomas. So this is a PET scan of a patient before he began treatment and then 8 months after treatment with this drug. And excitingly, just a few weeks ago now, this drug, which is called Vismodegib, received FDA approval and is now going into the clinic to be used initially anyway for treatment of metastatic basal cell carcinomas. So I think there's a nice link there between, so that's the disease credentials for my talk. There's this link between the discovery of this gene initially in zebrafish and the development of this drug almost 20 years later. Now, of course, in the intervening period, we've learned a great deal about the Hedgehog signal transduction pathway. So the slide that I showed you from 1993 was rather more simplistic than this one, which summarizes the way in which the Hedgehog signal is thought to be transduced. And without going into lots of details, it's clear that Hedgehog is an unusual protein. It undergoes this dual lipid modification, which requires that it is transported out of the secreting cell, and a process which requires this large transmembrane protein dispatch, which interestingly has a significant homology to its receptor patched. And then one of the key discoveries which has been made over the last 10 years is the importance of the primary cilium in the response of cells to Hedgehog. So a lot of the processing of the signal, that is the processing of the response of the cells to the signal, seems to go on in the primary cilium. And a lot of this work, it has to be said, has come from studies in mouse and also in human tissue culture cells. But what I want to do in the rest of this talk is also show you that the zebrafish has made some contributions, and I think will continue to make contributions. So actually, patched was identified. There was a rapid flurry of activity, in fact. So Matt Scott initially identified the vertebrate homologue of Drosophila patched in early 1996 or late 1995. And in collaboration with him, we cloned and characterized the first of 2 patch genes in zebrafish. And we use this to show that PKA, protein kinase A, which is a modulator of the Hedgehog pathway in Drosophila, has a similar role in vertebrates. So by blocking PKA activity, we were able to show that patched, which is a target gene of the Hedgehog pathway, so when the Hedgehog pathway is activated, patched transcription is upregulated. And by blocking PKA, we were able to show that patch became ectopically expressed. Now, unlike Drosophila, it turns out the vertebrates have 2 patch genes. Of course, it's not uncommon for fish to have duplicates of genes, but in fact, all vertebrates have 2 patch genes. And these are the mouse patch genes, the expression of the mouse patch genes, patch 1 and patch 2. And this is the expression pattern of the fish patch genes, patch 1 and patch 2. And I have to say, I have to hold up my hands to and admit to introducing some confusion in the literature, because we named this gene patched 1 on the basis that its expression pattern was more like patched 1, although actually we showed, looking at the sequence similarities, that in fact patched 2, as it was called then, is more like patched 1, and patched 1 is more like patched 2. So I hope I confused you now. So the key thing is there are 2 patch genes, but in fish, patch 2 is regulated more in the way in a way more similar to patch 1 in mouse. So interestingly, if you look at the difference between patched 1 and patched 2 in most vertebrates, the patch 2 is characterized by having a much shorter C-terminus than patch 1. But in a fish, the C-termini of both proteins are very similar in length and actually have significant levels of homology. And in fact, whereas in mammals, it's sufficient just to knock out patched 1 to get an effect which is equivalent to the de-repression of the Hedgehog pathway throughout the neural tube, and that leads to the ventralization of the entire neural tube, as indicated here by the expression of this gene NKX2.2. In fish, it's necessary to knock out both patch 1 and patch 2 to see a similar effect. Whereas in mammals, the knockout of patch 2 has very little effect. The mice are actually homozygous viable and fertile. So that's the first major difference I want to highlight between the fish and mammals. Now, our ability to analyze the Hedgehog pathway in the fish has really been helped enormously by the genetic screens which were performed in the 1990s by Mark Fishman and Wolfgang Drever in Boston, and by Jannie Nusslein-Volhard and her colleagues in Tübingen. And there were two sets of mutations which turn out to encode components of the Hedgehog pathway. One set were identified in this screen, which was actually done in collaboration with Friedrich Bonhoeffer, looking for mutations affecting retinotectile axon pathfinding. And in fact, the patch mutant I just showed you in patch 2 is this mutant blowout. And then the second group of mutations which were found, which are those which affect somite structure, or that's their principal phenotype. That was the phenotype on the basis of which they were identified. And these were called the U mutants. And a number of these are also U2, Sonic U, and Chameleon. And a key discovery in the late 1990s from Pascal Hafter's group was that Sonic U is the Sonic Hedgehog gene. And in fact, the last time I spoke at a Cold Spring Harbor symposium, Pascal, who is now sadly deceased, presented this exciting result in old Cold Spring Harbor in Long Island. And what he and his colleagues showed was that in the absence of Sonic Hedgehog, the patterning of the neural tube, which is indicated here by the expression of this transcription factor of FKD4, which is normally expressed in essentially a 3-cell wide stripe at the ventral floor of the neural tube, is affected so that these 2 rows of cells flanking the midline, which are known as the lateral floor plate, no longer form. And we went on to look at this same marker in all of these other mutants, which I've mentioned, and found similar phenotypes, suggesting that they're all involved that they're all encoding genes involved in hedgehog signal transduction. And over the next few years, we and other groups, particularly Rolf Karlstrom and Alex Scheer, positionally cloned some of these genes and showed that they encode the transcription factor GLI2, the dispatch protein, which is involved in the secretion of hedgehog, and other of the transcription factors GLI1, and then a co-receptor for the hedgehog ligand called BOC. And in addition, I want to highlight the fact that 2 other members of this class identified genes which have not previously been implicated in hedgehogs. So this was an original contribution of the fish to understanding the hedgehog pathway. This one, which is U, encodes a protein called SCOOB2, and this one called iguana encodes a protein called DZIP1. So those were all phenotypes which affect the patterning of the neural tube. In 1996, Pete Curry in my lab showed that hedgehog also has a role in the zebrafish in patterning the myotome. And specifically, it seems to be involved in specifying a subset of muscle cells called the muscle pioneer cells. And we went on to show that actually hedgehog is required for the specification of an entire muscle lineage called the slow-twitch muscle lineage, of which the muscle pioneer cells are a subset. And subsequently, we showed that the way in which hedgehog regulates the specification of this lineage is through through the regulation of a gene which was identified by the U-boat mutation. This is another member of the class identified by Janni Nusslein-Volhard's group, which we then showed actually encodes a transcription factor called BLIMF1, or now known as PRDM1. So the key finding here was that, as in the neural tube, in the somites, hedgehog is specifying different types of cells, slow-twitch muscles and muscle pioneers. And by using cyclopamine, which is an inhibitor, a pharmacological inhibitor of hedgehog signaling, we were able to show that different levels of hedgehog signaling are required to specify these different cell types. And these are the data here, which were published in this paper in 2003. So at low levels, hedgehog activates transcription of the PRDM1 factor, but it seems that at high levels, hedgehog is required to activate expression of genes which are specific to the muscle pioneer cells, and the most prominent member of that class are the Ingrail genes. And only fairly recently, a student in my lab, Ashish Maria, took an approach of looking for cis-acting regulatory elements which control Ingrailed, and identified this element here, which drives expression specifically in the muscle pioneer cells in response to hedgehog. And surprisingly, he found that this actually is under the regulation not only of hedgehog, but of also the BMP pathway, and that in fact the accumulation of phosphorylated SMAD, which is the activating transcription factor which is regulated by BMP signaling, correlates with the absence of transcription of this gene. In other words, where SMAD is not present, the engrailed gene is activated, and where SMAD is present, the engrailed gene is repressed. And that led us to this model, which I'm not going to take you through anyway, of crosstalk between the two pathways as being key to the activation of these engrailed in these cells. So let me say a bit more about the GLI transcription factors, and again compare and contrast with the mouse. So in the fish, originally one GLI2 gene was identified called GLI2. One GLI2 was identified by the mutation called U2. U2 turns out to be act as a dominant negative, so it causes a truncation of the protein, which acts as a constitutive repressor. So we used morpholinos to knock out the function of GLI2, and actually found that whereas in the total, in a U2 mutant where Hedgehog pathway is being repressed, we lose all of these cells. In a GLI2 morpholino, there's actually a slight increase in one class of cells. So GLI2 is acting both as a repressor as well as an activator. And then DTOR, which is a mutation in the GLI1 transcription factor, has relatively little, if any, effect on this process. But if we combine the two mutations or knock out both genes simultaneously, then we lose the specification of this cell type. And that has resonances with what had been shown by Alex Joyner and her colleagues in the mouse, where knocking out GLI2 leads to a slight defect in the diencephalon in this case. This is the expression pattern of NKX2.1. But removing GLI2 and GLI1 simultaneously results in the total absence of this cell type marked by this gene NKX2.1. But the difference here is that this function is all in the mesoderm. And in fact, the loss of GLI2 seemed to have no effect in the nervous system. And the explanation for that came a few years ago when Gong and Korsch in Singapore showed that there are, in fact, two GLI2 genes in zebrafish, one of which seems to be dedicated to the mesoderm, and the other which is expressed predominantly in the neurectoderm. And then knockdown of this GLI2B gene, as it's called, has specific effects on neural markers. Another difference between GLI2 in the fish and mouse is that so what I haven't actually explained very well, but GLI2 is a bifunctional, or GLIs are bifunctional transcription factors that can act both as activators and repressors, and they get transformed from being activators into repressors by their cleavage. And the cleavage is what is promoted by their phosphorylation by protein kinase A. So if you look in the mouse, then a protein called GLI3, encoded by a third GLI gene, is very efficiently processed into this repressor form, whereas GLI2 is rather inefficiently processed into this repressor form. In the fish, by contrast, we find that actually the predominant form of GLI2A is the repressor form. And this is regulated, as you can see, by hedgehogs, because if we look now in a patched 1, patched 2 double mutant, in which the hedgehog pathway is de-repressed, then there is none of this repressor form formed. And conversely, if the embryos are treated with cyclopamine to block the hedgehog pathway, then all of the protein is found in the repressor form. I'm going to move on now just to talk about other targets of hedgehogs. So I've mentioned a couple of genes, PRDM1 and engrailed, which are regulated by hedgehog in the myotome, but we wanted to look in a more unbiased way. So we've been using ChIP-seq, and this is work done by a postdoc in my lab, Wang Xingang, has been using ChIP-seq to identify targets of hedgehog. And he finds about 350 highly enriched peaks when he does these experiments, and this lists some of the genes. And the ones which are shown in yellow are the genes which we would expect to find. These are bona fide, well-characterized targets of hedgehog, namely patch2, GLI, and patch1. And the ones shown in red are ones which he has so far validated by a variety of approaches. And just to show you this is what the data looks like. So these are the peaks which we find associated in this case with patch1, and this peak corresponds to a highly conserved sequence throughout the vertebrates. And in fact, it was shown some years ago that this site, which is blocked here, which is a consensus binding site for the GLI transcription factor, if mutated, abolishes the expression of a reporter gene, which responds normally to hedgehog and tissue culture cells. In the case of patch 2, there is this peak here just upstream of the transcription unit, and if we take this fragment and hook it up to GFP, we recapitulate the pattern of expression of patch 2 very nicely. And so Ingham has been looking, as I said, he's been validating some of these other genes which have been identified through this approach and found a large number of them. This is just a few examples here to show you how we do the validation, which is to look at the wild-type expression, show that it's expressed in the cells where we expect it to be expressed, that it's absent in the absence of hedgehog, and it's overexpressed, it's ectopically expressed when the hedgehog pathway is ectopically activated. And none of these genes have previously been identified as putative targets of hedgehog. So another interesting question is how is GLI, what other factors are involved in the regulation of GLI? And clearly an important process in GLI activation is its transport from the cytoplasm into the nucleus. And again, a few years ago, when we were studying the iguana protein, the iguana mutant, we found that it encoded this protein called DZIP1, which had no previously known function, but it has this interesting structure of a zinc finger and several coil domains. And the iguana mutant has this interesting phenotype of causing an upregulation of the hedgehog pathway. So at that time, we proposed that iguana might be involved in somehow in restricting the nuclear uptake of the protein. And in order to study that, we actually tagged GLI2 with GFP and looked at its expression in the embryo. And then we were slightly surprised to find that GLI2 tagged GFP localizes very strongly to the primary cilium, which is something which had already been described in the mouse. And in fact, this localization is dependent upon hedgehog pathway activity, as you can see here. And what was remarkable was that in an iguana mutant, the primary cilium was very truncated. GLI2 still localized to the primary cilium, but there wasn't very much of the primary cilium left. And so this led to the realization that actually iguana is somehow involved in the morphogenesis of the primary cilium, and the iguana protein itself actually localizes to the primary cilium. Now, just before we published that, actually, Alex Shear had published a very nice paper in which he and his colleague had removed IFT88 from the germline of fish. Now, IFT88 is one of the intraflagellar transport proteins, which is essential for ciliogenesis. And it had been a mystery in the fish because whereas these mutants in these genes had been identified in mouse and shown to cause aberrant hedgehog expression, or response, these phenotypes were not associated with zygotic mutants of the IFT genes in fish. But by removing IFT from the from the maternal genome as well as the zygotic genome, they found that they got a phenotype very similar to this, which actually is a phenocopy of iguana caused by the maternal zygotic mutant of another gene which we generated called TALPID, which is another gene required for ciliogenesis. So it's clear from this that primary cilia are crucial to hedgehog signaling in fish, just as they are in mouse. And I think the other important thing that comes out of this analysis is the importance of removing the maternal contribution of genes from zygotic mutants. In many cases, it's going to be clear that a zygotic phenotype is going to be masked by maternal contribution. So I want to finish with just in this last section then by turning to what I call the enigma of fused, suppressor fused, and the role of COSTL2 KIF7. So in the figure that I showed early on, these two genes fused and KIF7 figured as key components of the hedgehog pathway in Drosophila. And so we set out to ask whether or not that these roles were conserved in the fish initially using morpholino knockdown. So we knocked down fused using morpholinos and showed that this attenuated the response at the embryo to hedgehog signaling because the expression of patched is downregulated and these cell types which require high levels of hedgehog signaling disappear in the fused morphant. But a couple of years later, these two papers came out describing the mouse knockout of fused, which clearly showed that fused has, in the mouse, or also known as STK36, has no role in hedgehog signaling, which was a bit of a surprise, and always a worry when you're using morpholinos. So we also, at the same time, had knocked down suppressor of fused, which in Drosophila is enigmatic because actually a complete deletion of suppressive fuse has no phenotype. It was only discovered through its ability to suppress the fuse phenotype, hence the name. But in the fish we found, but all of the genetics suggested that it acts as a negative regulator of the pathway. And in the fish we found that knocking down suppressive fuse has a phenotype which is consistent with it acting as a negative regulator, although nothing like as strong as knocking down both patch genes. So a couple of years later, we were again surprised when this paper was published by Rune Tofgaard and his colleagues, who showed that elimination of suppressor fused in the mouse causes a total derepression of the hedgehog pathway. And in fact, the phenotype of the suppressor fused mutant in the mouse is almost indistinguishable from that of a patch knockout. So here is, again, a marked difference between the fish morpholino data and this mouse knockout phenotype. And these authors suggested that there had been a divergence in the pathway, and that in Drosophila, the requirement for fused and costal 2, which is absolutely crucial, had somehow been lost, in vertebrates, and now suppressor of fuse had assumed the role as the main intracellular negative regulator of the pathway. But again, we were surprised by that because, as I said, we'd also been looking at the fish ortholog of costal, which is known as KIF7, and had used morpholinos to knock down KIF7 and had shown a weak activation of the pathway when KIF7 was knocked out, suggesting that the role of KIF7 as a negative regulator is also conserved between flies and fish. And this issue was taken up by these authors, Jussi Taipale and co, and I always like to quote this to my mammalian friends to show what we're up against in the fish world. So they say, and I'll read it, although mutants of fused or costal 2 orthologs of zebrafish have not been identified, recently Wolf et al. And Tay et al. Have reported that zebrafish homologs of fused and costal 2 act in the hedgehog pathway based on morpholino antisense injections. In contrast to these findings, mice deficient in mouth ortholog of fused do not have a hedgehog-related phenotype, and mouse orthologs of costal 2 do not affect hedgehog signaling. This report. Then they say, hedgehog-related phenotypes can be observed in zebrafish by morpholino-mediated targets of other genes as well, such as beta-arrestin. It is widely appreciated that multiple types of embryonic insults result in hedgehog-like phenotypes. Thus, it is possible that the zebrafish phenotypes observed are caused by the morpholino injection process itself. Ignoring the fact, of course, that the phenotypes are opposite. So we were pleased when Piao-Tien Chung, who had published the mouse knockout paper a few years earlier, published this paper in Nature a couple of years ago, which basically recapitulated the data which we had published with the fused morpholino. Although I will say something about that right at the end if I have time. And even more importantly, a couple of years ago, these 3 papers came out showing that actually knockout of KIF7 in the mouse also causes a derepression of the hedgehog pathway. So again, this function is conserved between mouse and vertebrates, and as had been suggested by the morpholino data which we'd published. And if you look at the distribution of the KIF7 protein, either in mammalian tissue culture cells or in zebrafish embryos, you see it's localized to the primary cilium in a hedgehog-dependent manner. So where hedgehog is activated, KIF7 accumulates at the tip of the cilium. In the absence of hedgehog, it's stuck at the base of the cilium. And here you can see in wild type 2 cells in different regions, one in which is responding to hedgehog and this one presumably which isn't. So just really to nail this, and this is the last part of my talk, we decided we would make a real mutation in KIF7 using zinc finger nucleases. We made this mutation, which causes a deletion, an introduction of a stop codon through a frame shift just at the end of the motor domain, so it should be a complete null for the protein. Made the homozygotes, and this is work done by 2 postdocs, Ben Jin and Ashish Maria, now a postdoc. They made the homozygotes are viable and actually look completely normal. And then when Ashish went back and crossed his engrailed lac Z in and looked very carefully, he could see this weak ectopic activation in larvae at about 3 or 4 days. So he then took these homozygous females and crossed them to homozygous males, and then found a pretty strong hedgehog gain-of-function phenotype, which is shown here. I won't go through all of the points, but I hope you can see that patched is ectopically expressed, engrailed is ectopically expressed, bone formation occurs precociously, and, but interestingly, in contrast again to the mouse, there's no effect, or very little effect in the nervous system. And this, actually, in the interest of time, I'm going to skip that and get to the important point. So again, just a few months ago, these two papers appeared, well, nearly 9 months ago now, actually, these two papers appeared reporting KIF7 mutations in human patients with fetal hydrolephalus and acrocallosal syndromes, in one case, and in a ciliopathy called Joubert syndrome in the other case. Now, the interesting thing here is that this child here who suffers from this syndrome is homozygous for this truncation of KIF7, which is very similar to the mutation we made in fish. So it's a homozygous viable mutation in human, whereas in the mouse it's homozygous lethal. And the other interesting thing is that there are other patients who are heterozygous but actually can show phenotypes as strong, if not stronger than this. So that suggests that there may well be some interaction with other loci. And so interestingly, Ashish took our KIF7 mutant, which this is the zygotic KIF7 phenotype, crossed it to iguana, and this is an iguana heterozygote, and now in the double mutant, or rather the KIF7 zygotic homozygote iguana heterozygote, we get this pretty strong de-repression of the phenotype. And so I think that this makes this a nice background in which to screen, either to test candidate genes found in patients, or even to screen for novel ciliopathy-associated genes. And just some recent data shows that actually all 3 of these proteins can be pulled down with an antibody against GLI2A. So, sorry, I've overrun slightly, but just to finish, what my conclusions are, I hope I've convinced you that the core components of the hedgehog pathway are conserved between arthropods and vertebrates, and that the primary cilium is required for the cellular response to hedgehog throughout the vertebrates, and not just in mouse, as had been thought until a couple of years ago. As I said, many loss-of-function phenotypes are likely masked by maternally deposited gene products, and that's illustrated by IFT88, TALPID, and KIF7 that I've described to you here. There are differences in the relative contributions of certain components of the cellular response between teleosts and mammals, and forward genetic screens in zebrafish have identified novel vertebrate-specific roles for DZIP1, which I've mentioned, and SCOOB2, which I haven't mentioned, but it's a very nice piece of work by Pete Curry, who's sitting there, and Phil Beachy and Will Talbot. The role of this, I should come clean and say that the role of fused still remains unclear, because that's the one case where we've made a mutation and can see no phenotype. I'm happy to talk about that, but I'll finish now and just acknowledge, I've mentioned a number of people as I've been going through, but the people who've been working on this most recently in the lab in Singapore are Ashish, Ben Jin, and Wang Xinggang, assisted by Ashley Ng. And Julius Muller in Ernesto Guccioni's lab has been really extremely helpful in helping us with the bioinformatic analysis of the ChIP-seq data. And the ChIP-seq data was generated by Yijun Ran in the Genome Institute of Singapore. And these are our co-collaborators in the UK, and these are the funding people. Thank you very much.
Before we start question, I just want to make sure you guys know where the microphone is. So we have a total 4 microphones around.
I got a question for the fused story, and you generate a zinc finger knockout. And so the phenotype is clearly different, you pointed out, between the neural tube and the muscle. The muscle phenotype is quite conserved as observed in other organisms. So can the difference between the phenotype difference between neurotube and the muscle be explained by the, say, different requirement for the dosage, or hedgehog dosage, or any other explanations?
Sorry, I should have told you at the outset that my hearing is really bad.
Sorry about that.
I'm not sure. Were you talking about the KIF7?
The difference, I mean, can you explain, what's your comments on the difference between like a neurotube, almost no phenotype on the neurotube?
In the KIF7 mutant?
No, the fuse, if I remember fuse. You generate fuse mutant, right?
The GLE?
The fuse.
The KIF, yes.
Fused.
In the fused mutant.
The fused mutant,
Yeah,
I beg your
sorry.
pardon. The fused mutant, I didn't actually show you the fused mutant. I showed you a fused morphant.
Yeah, okay.
And the fuse, as I said right at the end, we have actually made a fused mutant, which has no obvious phenotype anywhere. So I really can't comment on that in any sensible way because it's not clear what the morphant phenotype really is.
Okay, thanks.
Lynn.
Phil, I had a question about the ChIP-seq data that you did. Could you tell us a little bit more details about how you did it and when you did it?
Yes, sorry, so I whizzed through that. So you saw two traces. So we purified, they were taken from whole embryo extracts and then precipitated with a polyclonal antibody against GLE2A, Zebrafish GLE2A, at the 5-somite stage and the 15-somite stage.
Did you see have you ever done any other ChIP-seq to see if there were co-binding? Like one of your stories was about how GLE and BMP will co-bind.
Yeah, so we've done that on individual, we've not done ChIP-seq for SMAD, but we've done that on that specific promoter fragment and shown that SMAD actually binds to that. Yeah, yeah. Oops.
Question? Phil here. Actually, I have one of two. So you mentioned that Hedgehog and BMP interact in the mitome, right?
Yeah.
At Ingram's gene. So do you notice they have any other cell types also?
Well, that is something that has been reported previously in the mammalian neural tube, of course, this interplay between BMP and Hedgehog. So I don't think it's completely novel. Again, our analysis at this point has been restricted to the myotome simply because we have the tools for doing it. But I think what is novel is the observation that Hedgehog, which again I didn't have time to talk about, but that Hedgehog itself is modulating the BMP response. So it's not simply an intersection, an integration of two pathways, but actually one pathway is the activity of one pathway is dependent upon the other. But that would take me another 10 minutes to explain properly. So.
Okay. If there are no more questions, then we we'll move to the next one. So our next speaker is Andy Miller, who is from Hong Kong University of Science and Technology. Today he's going to talk about some story about calcium signaling.
Well, I'd like to thank the organizers for inviting me to talk, but not to talk after Phil. It's like following the Rolling Stones onto the stage. But today I want to tell you a little bit about some work we're doing in the lab. And it involves calcium signaling during muscle development. So there's quite a bit of a history in this, looking at signals in cultured muscle cells, and indeed not just in the cytoplasm of the cells, but there's also good evidence that, we have nuclear signaling as well as cytoplasmic signaling, and you can also accelerate the differentiation of cultured muscle cells by electrical pulses, by releasing calcium into the medium. And this has gone on into intact systems. This is some early work by Nick Spitzer's lab, looking at myofibril differentiation. Here's another nice paper by Caroline Brennan's lab, and also we've published a couple of things about this. But I just want to emphasize that zebrafish are a wonderful model to do imaging studies in, especially calcium imaging. And as we'll hear throughout the week, they're very good for doing disease modeling. So just for those of you who don't work on muscle, just to tell you that there's two types, two basic types in zebrafish. You've got these slow muscle cells on the outer periphery here of the trunk, and here's the fast muscle cells that develop slightly later. And this gives you just a timeline here. So around 17 hours post-fertilization, these contractions start, and then here you get about 21 hours, you get this response to touching the head. Now, just to let you know very briefly, I'm going to whiz through this first part. My lab uses aquarin to look at calcium. It's a bioluminescent protein from jellyfish. It's actually a protein complex between a protein here and a small prosthetic group. And the whole, quickly summary here, it reacts with calcium ions to give blue-green light. And here's a brief schematic. How do you image with calcium? You basically load your sample with aquarin. You either inject it or you can express it. Very important is the amount of light that's generated is very small, so you need an extremely sensitive camera, and here you get a readout. And the point is aquarin imaging is excellent for low-resolution imaging over extended periods of time. So if you inject the embryo with the whole of aquarin, it's basically used up just about the time that the muscles are forming, which is a bit of a bind. But you can overcome this by either injecting them with mRNA to transiently express it, or you make transgenic fish lines, and then you basically incubate the embryos with the sealant and you get the aquarin complex. Nice thing, the gene's been cloned, so what we can do now is we can target the aquarin to different tissue types. So we've made a transgenic fish line. We've used an alpha-actin promoter to just put it in the skeletal muscles. So we have this lovely model fish to look at calcium signaling from a specific tissue type. And what you basically get are these nice signals. This is around 18 hours post-fertilization. We'll look a little later on. The signaling changes. And when you actually look at it over time, we get these lovely profiles. There are two signaling periods here. We call this SP1. And then there's a quiet period in SP2. And what's really interesting about these signals is the frequencies. So this early signaling here is quite low frequency. It then changes to higher frequency signaling. This is in the SP1. And why we're interested in this is that these signals, we think, encode information, so you can get both frequency modulation and amplitude modulation. So this is how these signals are read in the cells. These signals are extremely robust. The black boxes here are from a transgenic fish. These are from injecting mRNA. They look really good, and if we compare them, this is Caroline Brennan's work using a fluorescent reporter. Again, the frequencies are extremely consistent, and also in the somites that they're actually generated in, we hardly ever see any signals in somite number 1. They're generated in up to about somite 12 in a very, very predictable fashion. So where do these signals come from? We've poor resolution with the aquarin. So Phil mentioned earlier on cyclopamine or forskolin. You can knock out the slow muscle cells. You get rid of the signals. Or we take a genetic approach and use this mutant. There's no slow muscle cells, no signals. And then how are the signals generated? Here we've treated it with 2-APB. This knocks out the IP3 receptor. And immediately we have treatment. We see the signal. We take a tail cut, put them in the detector. It knocks down the signal immediately. Whereas if you do the same thing with ryanodine at an inhibitory concentration, this is knocking down the ryanodine receptor. Again, it's not immediately knocked down. So this leads us to think that the early low-frequency signals are generated with IP3 receptors, where the later signals in SP1 are generated mainly by ryanodine receptors. And the SP2s, I'm not going to really talk about them today, but they seem to be mainly ryanodine receptors. And this is borne out by looking at the expression of the receptors within the cell. If you look in the muscle cells here, if you look in SP1, it's mainly IP3 receptors that come up first, with very little ryanodine receptors being expressed at this time. If you knock down these signals, let's have a look at what happens. So here's the controls. Surprisingly enough, there's not a lot of effect of the knocking down the IP3 receptor, but there's a dramatic effect of knocking down the ryanodine receptor. We get these, it looks like a release of the slow muscle cell from the vertical myoceptor, and you get these patches here of actin and myosin in the cells. So we don't have much resolution, so we go to confocal fluorescent microscopy. Now that we know where the signals are generated, we can go and look at the SP1 signals. And what's really interesting is these signals early on are mainly nuclear with a small cytoplasmic component. And as I said, the frequencies are quite interesting. So this is this early phase in the SP1. If we go on a little bit later in SP1, there's still a nuclear component, but a lot more cytoplasmic component to these signals. And again, across the myotome blocks here, they signal together. And also, if you look from dorsal, medial, and ventral, they're all firing together. Again, the SP2 signals, I just want to say here, there's very little nuclear component. These are mostly cytoplasmic, and think these are functional signals as the muscles contract. So again, to put this into summary, this is Caroline Brennan's results. Here's ours, our SP1, our quiet period, and here's our SP2 period. It's quite interesting because these contractions, they correlate with the expression of dystrophin and the vertical myoceptus. So here's the interesting new part of my talk, is this is this new messenger that's recently, it's been known for some time, but now that it used to be called the NAADP receptor, now the receptor's been identified as this 2-pore channel in the acid vesicle, and it's very important. This is the most sensitive calcium mobilizing agent that's been discovered so far. It can release calcium at nanomolar levels. The calcium that's released through the 2-pore channel is known to affect these other ryanodine receptors and the IP3 receptor. And the idea is this works as a little trigger mechanism, that it can release a little bit of calcium that then activates either IP3 receptors or ryanodine receptors in the sarcoplasmic reticulum, and then you get this calcium wave. And again, there's 3 isoforms in the early endosomes and the lysosomes. So this brings us to this collaboration with the group in Oxford. They've shown in muscle culture cells and also in C2, C12 cells that if you upregulate NAADP, you can increase the differentiation, what they call the differentiation index. If you block the channel, you reduce it. Their idea is, here's this idea that they're a little trigger, and they've showed quite nicely with lysosome tracking and bodipiryanidine that they have this nice association. And again, just for the disease part of the conference, there's around 50 diseases. 8 of these have been specifically linked to defective calcium homeostasis in the lysosome, and we're quite interested in this Niemann Pick disease. So what we try to do now is use the intact zebrafish. So here is this TPC2 morpholino, and what's quite interesting, it doesn't knock down the signal like IP3 or IP3 agonist or ryanodine receptor antagonist does, antagonist. What it does is it modulates the signal. So it knocks it down in these early low frequency, what we think are IP3 receptor generated signals, but you can see it pushes the SP1 way beyond the normal level. So if you look here, this is comparing it. Here's the 2-APB, knocks it down completely. Ryanodine knocks it down completely. Here we've just got a modulation. So what does it look like? A little higher magnification. Just knocking down this TPC2 channel completely destroys the sarcomeric organization. Again, if you compare this, so we think we've got a very interesting story coming on here. We're looking at all the different sarcomeric proteins in line. This is alpha-actinin. It's quite interesting. The alpha-actinin bands are still there. We've done metamorphic measurements to look at the differences between the controls and these TPC2 knockdowns. If we look at the behavioral effects here, again, this is where they twitch. This is where the head touch begins. If you very quickly, I don't know if I can start this movie from here, but maybe the guys can start it up here. But here's the TPC injected morpholino embryo. There's absolutely flat, no contractions at all, whereas here's the control embryo twitching quite nicely. And again, the same with these head touching. The morpholino injected embryos don't respond at all. So here's a model that we've published recently, and now we have to add these TPCs, possibly at the nuclear membrane, triggering nuclear calcium release, because these is where these early signals are seen, and somehow or other we haven't quite worked this out yet, how these signals result in the sarcomeric organization. So my final couple of slides. So why do these embryos twitch at this early stage? Well, we believe they need to twitch in order to build up proper muscle structure. And just finally to acknowledge, this is the group, Jeff, Sarah, and Mandy in my lab at HKUST. And these are our collaborators in Oxford at the pharmacology department, mainly John Parrington, Anthony Gaglione, and also our collaborators in UCL, David Whitmore. And this is funded by the Hong Kong Research Grants Council. And my last slide is just to say, to give you a pre-invitation to Hong Kong next year. We're going to run the 6th Asia Oceanic Zebrafish Conference sometime probably in August. So thank you very much.
Questions?
Sorry to run through it so quickly. My talk's going to be available if anybody wants it, or they can come and talk to me in the bar over a beer, and I'll be happy to go a bit slower.
So the sarcomeric collapse looks like it's basically, as you described it, there seems to be two different forms, one reliant on myosin deposition and one on actin. Is that true from what the slides you showed, or is it just you haven't worked out the primary defect there yet?
I really didn't have time to go through it in detail. It seems that the myosin, the structure of the myosin thick filaments appears to be more disrupted than the actin thin filaments. And we were quite surprised to see the alpha-actinin in the Z lines still seem to be being expressed. But we're trying to use the proteins in the sarcomere as a readout in the intact fish. It's probably a crazy thing to do. It's trying to know if the drugs are actually getting in. We tail clip them and we put some fluorescent marker in the drug solution so we can see it going up the trunk of the fish. But it's always very kind of, is the drug there in enough concentration to actually have this effect?
I assume these things are still over here.
Yeah, sorry.
Oh, I'm still talking to you, Andy.
Oh, sorry. Sorry, I haven't even had a beer yet. It's coming out over here. Sorry.
We will, don't worry about that. So the T-tubules, are they normal in these things? One assumes that these proteins are localized there, and therefore is there a structural role of T-tubules inside?
We haven't had a look at the T-tubules yet. That's a very good thing. And also, we haven't even looked at the SR to see what it looks like yet. But that's certainly something we're going on to do.
Sorry, this is a naive question, but when the tail wags one way and then the other, does the signaling go at one side and then the other?
That's a great question. What we really need to do with that is to look at the fish from a different angle. Yeah. There's no resolution in the z-axis using a corin. So the light's generated from anywhere within the three-dimensional trunk of the fish. So when we look at it from the side, you're right. I think the idea is it twists one way and then the other way. So you should get alternate contraction on either side of the midline. But I think we need to look, with a corin, I think we need to look at it from the dorsal view.
Side. Yeah. The other question, inspired by if you look at myocardial infarction in humans, there's a lot of disorder. And I'm wondering if there's any relationship, and then if whether or not there is, whether what you see in the disruption of the structure is reversible.
Yes. Again, that's something we really would like to look at, but we haven't done so yet.
I saw a very nice picture of the calcium signal. My question is your picture from the transgenic fish line you make? It's named AEQ or just regular calcium signal kit?
Okay, that's, we deliberately targeted the acorin to the skeletal muscles using an alpha-actin promoter. But we could basically, if anybody's interested in using this technique, you can target the acorin to whatever you really like in the cell, or not only just different tissue types, you can target to intracellular compartments as well. So you could target it to the nucleus or to the mitochondria or any other part of the fish. It takes a while to do it.
Okay, thank you.
Any more questions? So if there are no more questions, let's thank Andy. I think we can have a 5 minutes tea break. Yeah. So, and now we come back in 5 minutes. Right.